Dear learners, I welcome you to the second lecture of RNA according to your syllabus. And the first lecture was focused upon the structure of RNA and its basic instability. And this lecture is focused upon the first type of RNA, that is the messenger RNA. Before going in the detailed structure of messenger RNA, we must first understand that all the three types of RNAs, as in the last lecture you have already studied, that there are three types of RNAs: messenger RNA, transfer RNA, and ribosomal RNA. Okay, so all the three types of RNAs are basically involved in one process that is called as protein synthesis, or it is also called as translation, right? So the protein synthesis process starts from the DNA, and the DNA has the information regarding which protein is to be synthesized, and this information is passed to the messenger RNA. So this is the RNA molecule whose main function is to take the message from the DNA that which protein is to be synthesized and to deliver this message to the ribosomes. Okay, so now I think you have an idea that what messenger RNA does. It takes the message from the DNA and it delivers the message to the ribosomes. Okay, now it takes the message and delivers actually means that this messenger RNA which we are talking about, okay, is a complementary copy of DNA. Got it? That what is a messenger RNA? It is a complementary copy of the DNA, right? And the process of synthesis of messenger RNA is called as transcription, right? So here in the figure, you are seeing two RNA molecules. Let us first understand the first RNA molecule. The first RNA molecule is present in prokaryotes, okay, and this structure is present in eukaryotes. Now let us first understand in prokaryotes. In prokaryotes we find that there are multiple start sites and stop sites. Okay, here you can see start site, stop site, start site, stop site. Okay, so one start and one stop site is involved in synthesis of one protein. So here you are finding that this RNA is involved in synthesis of how many proteins? Three different proteins, right? And therefore such kind of messenger RNAs which contain messages for synthesis of more than one proteins are called as polycistronic. Okay, what they are called as? They are polycistronic. And those RNAs which, I'll just complete here. Okay, so the spelling is completed over here, polycistronic. So such messenger RNAs are called as polycistronic RNAs, and in contrast the eukaryotic mRNA, you are seeing that there is only one start site and one stop site, and that is required for synthesis of only one protein, right? So such messenger RNAs are called as monocistronic. So you get the term, either the mRNA can be polycistronic or it can be monocistronic, right? Then we come to the structure. Every messenger RNA has one 5' end and it has one 3' end, okay? and there are no base pairings present in any of the part of messenger RNA. It is a totally linear molecule. Okay, so what is the main structural peculiarity? That the messenger RNAs are linear in nature. There is no base pairing, right? Now at the 5' end you will find that there is a phosphate and a guanine residue is attached at the 5' end, and that too in a reverse orientation, and the bond is between 5' 5'. So this is a very typical feature, and this 5' end is called as the cap, right? At the 3' end there are many adenines present, and that is called as a poly A tail. Over here it is signified, okay, and this tail is present at the 3' end. Now in between the 5' end and the start site, there is a site which is called as ribosomal binding site. Okay, so this is the site which is shown in the purple color. You see here it is marked, here it is marked. It is a site which binds with the ribosomes. It helps the mRNA to bind to the ribosome, right? And in case of prokaryotes, this ribosomal site binds to the ribosomes for attachment. Okay, now in case of eukaryotes, there is only one starting point and one stopping point. Okay, at the starting point you see the sequence is AUG, that is a start codon is present, and at the end a stop codon is present. Here the stop signifies codon, right? So there are codons which help in starting and stopping of the protein synthesis. And what are codons? Now for this topic we will limit what codon is. 3 nitrogenous bases together which has ability to code for a amino acid is called as codons, right? A brief overview of what we have just seen in the figures. First thing which you have to understand is there are modifications in the messenger RNA at 5' end and 3' end. Okay, and there is a cap present at the 5' end due to reverse binding. The function of the cap is it helps in bringing or recruiting the ribosome close to it. Okay, after attachment, the ribosome, it moves through from 5' end to the 3' end till it finds the AUG or the start codon. Okay, this process is called as scanning, and just 3 bases upstream of guanine and immediately downstream of 3 bases upstream of this is AUG. Okay, upstream of the start codon and just before the downstream of guanine. Upstream means before, a sequence before that. Okay, and downstream means a sequence after that, towards the right side, right? So in between the start codon and the G, there are base sequences which were first of all analyzed by Marilyn Kozak, and therefore those sequences are called as Kozak sequences. Kozak sequences are generally present in eukaryotes, but they are not a compulsory feature, and it is found that if they are present, they increase the efficiency of the translation, right? In some eukaryotic RNAs they may be absent also. The function of poly A tail is it helps in recycling of the ribosome as well as it helps in increased level of translation, right? So these are the major functions of the structures which we had studied in the messenger RNA. A messenger RNA is a slightly unstable molecule. It is synthesized after its function is over. It is destroyed. It is not stably present inside the cell. Okay, so with this we complete the structure of messenger RNA and its function, and in the next lecture we will understand the structure and functions of transfer RNA. Thank you.